So, a very good evening to one and all present here. I am Srijan Sen, and today I will be taking your week 10 live class session on the course Fundamentals of Protein Chemistry, which is being taught by Professor Shagata Das Gupta of IIT Kharagpur. So, I am sharing my slides with you all. So the last class what we discussed about is more about proteins, the different types of proteins, the membrane proteins and all. Today we will be getting into a little bit about nucleic acids and the different types of nucleic acids. How do we make these nucleic acids? What are the ways of engineering of these nucleic acids? And also, today's class is mostly about DNA, RNA, and the different types of nucleic acids that are known. So whenever we talk of nucleic acids, the first thing that comes to our mind is the DNA, that is the deoxyribonucleic acid. So DNA is a very small molecule made up of these nucleotides. Okay, so each nucleotide has three parts. It has a sugar phosphate. It is made up of sugar phosphate, a molecule that makes the backbone, and a nitrogenous base attached to it that has hydrogen bonds between them. So the nitrogenous base is a part of the nucleotide that carries the genetic information. So the bases found in DNA are adenine, cytosine, guanine, and thymine. So on the based on that, we know it as ATP, CTP, GTP, and TTP. Okay, so adenine forms base pair with thymine, and cytosine forms base hydrogen bonds with guanine. So a gene is a stretch of DNA that codes for a particular type of a protein that has a function in an organism. It is the unit of heredity in a living organism. So all living things depend on genes. Genes hold the information to build and maintain the organelles or the organism cells and pass the genetic traits to the offspring. So this is a very important thing, that is the gene that made up of DNA, and that will code for the different types of protein. So the message is converted in the form of a protein. It's expressed in a particular organism. So there is expression of these proteins that revolves around carrying out the different types of functions. So what are the basically the gene components? So the gene components are exons, that are a set of coding regions, and the introns, the non-coding regions, removed sequence, and therefore are labeled as split genes or splicing. Okay, so you have the exons that are basically the coding regions, and the introns that are the non-coding regions. So these introns, so why do we have these introns? Because so upon splicing is basically the protein becomes functional. It is because if only the exons would have been present, then there would have been excessive production of that particular protein, and then there would be uncontrolled growth of the protein, leading to different types of diseases. Specifically what happens is that there is cancer progression. So therefore these introns are present in between the small exon regions, such that this can be the control such that there is a control between the exon and the introns, followed by splicing of the exon regions, and ultimately these exon regions are responsible for the splicing activity. So therefore exons and introns are together very important for the function of a particular DNA or a gene construct, because only the introns or the exons might not be absolutely okay, right? So therefore, you together. So together, the thing is actually very required. Now, genome is a little bit different from that of a gene. So it's a combination or a complement of an organism, including its genes, that are represented as a genome. So genome is basically, it is a for kind of a library. So this library has different types of DNA in it. So it's a combination library of nucleic acids or DNA. So gene expression is a process by which the information from a gene is used in the synthesis of a functional gene product. So the functional gene product is basically a protein. So what you have a DNA, that is, so a gene is responsible for carrying an information, but for carrying an information or for storing an information, the gene is very, very important. But to carry a particular information or for the production of that particular function within an organism, you need it to be transcribed to mRNA, followed by the translation to the following protein sequence. So therefore the gene is actually transferring the information in the form of proteins. So the process of gene expression is very common in eukaryotes, viruses, prokaryotic proteins, and to generate the macromolecular machinery for life. Okay, so the different steps of gene expression are as follows. Like, you have the transcription of the mRNA synthesis, then is post-transcriptional processes in the form of RNA splicing. So why RNA splicing is required? Because, like I was telling, there should not be any uncontrolled protein expression within a particular organism. There has to be controller regulator. So these introns are basically the regulators to prevent any kind of overgrowth or over translation. So therefore splicing is required, because only when the protein wants to produce, it has to just mRNA, there is RNA splicing. Only the exon regions will be responsible for coding the sequence and for the production of proteins. Then you have the translation, that is protein synthesis, and the different post-translational modifications to carry out its function. So although there are different genetic changes that can occur within a particular organism or a particular system, like an alteration in the segment of a DNA which can disturb a gene's behavior leads to a disease. So one common example you have always heard of is basically the sickle cell anemia or the sickle cell RBCs. Okay, so there is a normal RBC, and just for the mutation of one nucleotide, just one particular nucleotide, the entire system gets disrupted. So that is what happens when there is a particular change in the DNA sequence that can lead to disease. So small changes are basically called as mutation. So there is a small genetic change, like a single nucleotide change or a silent mutation that is happening. So that's a mutation leading to a genetic drift. So there can be a certain drift, but genetic shift occurs when there is recombination, where there is a large segment of DNA or a large portion of the DNA gets changed and gets actually hampered. So that's the genetic changes that can occur within an organism. Now, mutation, as I was telling, is a change in a particular nucleotide sequence that can happen. Like, you have a CTT sequence in the wild type hemoglobin, it suddenly changes to a CAT. So what happens over here is that this codes for a particular amino acid. So GAA will code for a particular amino acid, and then you have GUA. So GAA is basically coding for glutamic acid, and suddenly it changes to GUA that codes for a valine. So just one nucleotide change can lead to complete disease condition that we do not want, but it does happen within DNA. It's very common. So, or mostly in diseased conditions, these are the changes that you might be observing. So, that's the wild type hemoglobin DNA versus a mutant hemoglobin DNA that you can see from the image, and the coding mRNA strand leading to the formation of a normal hemoglobin with glutamic acid and a sickle cell hemoglobin with value. So we know a little bit about mutations and all. Probably if you are there in a molecular biology lab doing some molecular biology experiments, you have heard of the recombinant DNA technology, cloning, mutation, site-directed mutagenesis. Okay, so you have known about that. But recombination is basically it occurs with time. So there is a natural occurrence of recombination, or there are certain viruses that incorporate these kinds of recombinations to be present inside the host, and also like you have the exchange of corresponding DNA segments between adjacent chromosomal material during the special type of cell division, giving it a new genetic makeup. So there is a particular segment, like a bigger stretch of DNA is actually transferred, leading to a chimeric protein or a chimeric transition. So there is removal of one portion of the DNA and it goes and attaches to the other portion, and that leads to certain kinds of chromosomal translocation, and formation of a chimeric protein might occur. So, like I was telling, so recombinant DNA. So that's about playing around with DNA. You engineer the DNA the way you want. You produce the proteins the way you actually are wanting to work in a lab or in a particular system. So recombinant DNA technology is the engineering of DNA. So why recombinant? Because you're changing the original sequence of the DNA and giving it a new sequence, and that also in a lab environment. It's not naturally selected. You are force incorporating that. So you can do that using different methods, like you have the molecular cloning. You can go for like site-directed mutagenesis. Then there is different types of techniques that you can incorporate to carry out this engineering of nucleic acids or engineering of proteins. So protein engineering will have all of these certain steps that are present. So it is possible, means recombinant DNA is possible because DNA molecules from all organisms have the similar chemical structure. So we know that there is an ATGC, so it's there in everything. When it's RNA, it becomes the thymine becomes uracil. So we know how different the nucleotide sequence are. So we have an idea about this nucleotide sequences, and we can just is play around with this sequences. So rDNA is a general name for a piece of DNA that has been created by the combination of at least two strands. So rDNA molecules are sometimes called as chimeric DNA, like I was telling, because of recombination that can happen. Now because they are made up of two or different species, we call it as a chimera. So chimeric proteins you can prepare as well. Otherwise, there are certain conditions where these chimeric proteins occurs naturally or in disease conditions. So recombinant DNA technology, what we need to do is isolate, clone a single copy of a gene, and we can make innumerable number of copies of that particular gene by a special technique known as the you people have heard of that. That's the polymerase chain reaction. So all are identical copies. Now what you do some kind of treatment with different restriction enzymes or some other types of enzymes and all. So some endonuclease enzymes, and you treat it upon the library of that copy, so the multiple copies of the gene, and then you put it in a particular vector and let it multiply in the bacteria or different expression systems. Okay, so when you are expressing in different systems, you will can find the multiple copies of it, and that, so the entire process of like uh, amplifying these genes and treating it with endonucleases, putting it in a particular vector and ultimately letting it to grow in a bacteria and getting like multiple colonies or allowing the cell to multiply and propagate is known as the recombinant DNA technology. It's very, very important. So like the endonucleases or the exonucleases that I'm talking about is basically the restriction enzymes. So they cut the bacterial enzymes that cut long strands of nucleotides into smaller segments. The beauty about how specifically these restriction endonucleases work is because they cut at specific sequences. They do not cut randomly, like they go and cut at any random point. That does not happen. So there are restriction enzymes that identify specific sites where they actually cut. And the beauty about these sites is that they should be palindromic in nature. Like if, for example, you have GAA-TTC, that's the eco-R1 site, the very common site that is known to all of us. The GAA-TTC, you see the reverse strand also from the 5' to the 3', it reads GAA-TTC. It's very much unique among these sequences because there are only a few specific sequences that can occur in such forms, and that is why the restriction enzymes are designed in a manner where they can identify these palindromic sequences and cleave them or chop them. So they recognize the specific sequences of nucleotides or bases and cuts the DNA at the specific site within the sequence. So one chain runs left to right and the other chains runs right to left. So that's the 5', 3', and the 3', 5'. So that's very common. So okay, now so single chain tails of DNA called the sticky ends are created, and that's the action taken by the restriction enzymes. So restriction enzymes not only prepare the sticky end, so the sticky ends are the ones where you get like this tail, long tails hanging over there, but it is also that it can cut in the center. Okay, so there are certain restriction enzymes that can cut in the center and they create the blunt end, then it becomes very difficult to actually join them. Okay, so it becomes very, very tricky in those conditions. The sticky ends readily bind to complement chains of DNA, and then there is phosphorylation and ligation, leading to the joining of the 3' OH with the phosphate group of the in the 5'. Okay, so that is how ligation works. So now you have to the pieces of the DNA have been cut with the same restriction enzyme that can bind together to form a new sequence. So restriction enzymes can be used to isolate or out a specific gene. Now, when you have treated your construct with, say, a restriction enzyme or something, you need to put it into a particular vector. So that's the cloning vector. So a DNA carrier that is used to clone a gene and transfer it from one organism to the other is known as a cloning vector. So once the gene has been isolated, that small piece of DNA can be placed into a cloning vector and can be introduced in organism. Now, many bacteria contain a vector called plasmid. So we know about, so these are the plasmid vectors. You have the DNA fragment. You enzymatically treat this with restriction enzymes, the vector as well as the insert, and you allow it to ligate and let it grow in a particular bacterial cell. So what is a plasmid? So plasmid is a ring of DNA, a circular coiled DNA, closed circular coil DNA found in bacteria in addition to its main chromosome. So there is a chromosomal DNA in the bacteria. Apart from that, you have the plasmid DNA that is also present. So it can be used to transport genes from one organism to the other. Okay, so that is the role of plasmid DNA. Now, for the process of using a cloning vector is that you need to have a plasmid, you need to be removed from a bacterial cell, so that the plasmid isolation. So what you do, you grow the bacteria in culture, and then you spin it down, you isolate it by treating the cells with different conditions like alkaline hydrolysis, and then you allow the composition to be in neutral medium, and then you will see that the chromosomal DNA gets entirely um, precipitated, and only you get in the soluble fraction is the plasmid DNA. From there you isolate the plasmid. Okay, so after isolating the plasmid, you treat it with a restriction enzyme as similar as that of the insert. So you're cutting it with EcoR1 and EcoR1, and then also you're cutting over here with EcoR1. So what will happen is basically it will allow it to join. Okay, so you have to cut the vector and the insert with the same restriction enzymes on the either end. So the donor gene, so you have the donor gene, a specific gene isolated from another vector. Now the plasmid is returned to the bacterial cell where it replicates as a bacterial cell device. Now this clones the donor gene. You can see the procedure. You have the plasmid vector, the DNA fragment to be cloned. You can see over here. Then enzymatically inserted, and then you mix it in E. coli cells. You make it the cells competent enough to take up this plasmid DNA. Then you allow it to replicate. So one cell can take up more than one plasmid. You get multiple colonies. You take them and again grow it in a culture, like maybe an LB broth or something if you're doing with bacteria, and then you let it isolate the plasmid in the similar way as before. So the bacteria contains clones of the donor gene that can be used to infect other organisms and transfer the gene to them. Okay. So now what happens in some cases that the plasmids are used to clone a specific gene so that the bacteria will produce a specific protein. So that is what we do in rDNA, is that we are engineering this particular gene construct. So this insert is very very important to us. We engineer this particular gene in such a manner that we can play around with this gene. So we can go for like a different type of a cloning. We engineer, we change the nucleotide, we mutate the nucleotides and all, and we do it. So for example, insulin is a protein that controls sugar metabolism. If you want to probably stop the insulin from acting, you mutate its active site residues. Maybe there is a serine, cysteine and a threonine over there, so you mutate these three residues to with alanine or anything like maybe glycine or something with very very different amino acid apart from the active site residues. And you will see what happens is that those particular a different kind of a protein will be produced that is not insulin, and it acts differently. So the process of rDNA is very simple. It's I have put it up as a step. So that our DNA fragment containing the DNA sequence to be cloned is known as the insert, that is isolated. You cut the DNA, then you join it using a ligase enzyme. So the insertion of the DNA fragments into the host cell is called a vector. Now the rDNA molecules are generated when the vector self-replicates in the host cell. The transfer of the rDNA molecules into an appropriate host cell is very very critical for the expression of the protein and the growth of in a particular bacterial system or an eukaryotic system, whatever system you want to grow it in. And then the selection of the host cells is very very important using a marker. So for like that we have the two types of markers. Usually the first one is a what do we call it, it's a colored marker for a generation of a color, or even an uncolored or a different antibiotic type of a marker. So do we have it okay, if I get it I will tell. So maybe okay, so then is the replica, so there is a selection. So one is like antibiotic resistance, the resistance marker, or colored marker. Okay, so it generates color, you can identify. So okay, so this is what my construct looks like. So replication of the cells carrying rDNA molecules to get a genetically identical cell or clone. Now for isolation, you need to isolate the vector. So the procedure of obtaining depends on the nature of the vector. Bacterial plasmids are commonly used vectors, and these plasmids must be purified from the genomic DNA. Now ultracentrifugation for extracting the plasmid DNA. So plasmid DNA is from distinct bands after ultracentrifugation in a gradient condition using cesium chloride and ethidium bromide. Now the plasmid band is collected by punching a hole in the plastic centrifuge tube. So this is the process of plasmid isolation. Then it's alkaline lysis. So what will happen over here? So in alkaline lysis there is HDS and sodium hydroxide. So these two are very very important because HDS is important in charging of the bacterial outer cell, due to which there will be repulsion and breakage within the cell. So the lysis of the cells occurs, and then the alkaline condition, that is NaOH or KOH, what it will do is basically it will open up the entire DNA sequence. So the DNA sequence remains like tangled, so it will open up everything, and even the chromosomal DNA and the plasmid DNA both gets disrupted or unfolded. Now what will happen, you have to fold them, but chromosomal DNA once unfolded takes a long, very very long time to fold again. So we do not need the chromosomal DNA, what we need is the plasmid DNA. So the plasmid DNA, we take it by just adding a neutralizing buffer of pH maybe low, so that of a low pH, such that the alkaline condition is removed, and then we take the plasmid DNA and isolate it. Then is the cutting of the DNA using endonuclease enzymes, like to generate the sticky ends that will allow for the formation of a linear molecule with the sticky ends. Now it identifies the sticky ends, and then comes the joining or the insertion, that is via ligase enzyme or the DNA ligase. A vector in any DNA molecule is capable of multiplying. So ligase is very very important for joining the 3' and the 5'. So the 3' OH with the 5' phosphate groups get joined via this DNA ligase with the help of ATP, that is the source of energy of this ligase enzyme. And then what you need to do, you need to introduce the plasmid or the vector DNA into the host cells. So that is via transformation. So how you do it, you take the plasmid, then you make the E. coli cells or whatever bacterial cells as competent by making it electrocompetent or by salt competent, like calcium chloride, then you have rubidium chloride and so on. So what are these ions important? So they are important in opening up or making like holes within the uh, cell wall. So okay, so to allow the plasmid to get into the inside the cell. Okay, so that is what happens over here. So it allows holes to appear briefly in the cell surface membrane of E. coli, making it permeable to DNA, and the plasmids does enter, and then you add uh, put it in ice to just block the membrane such that the plasmids do not exit the from the inside. So there is another way, that's the phage vectors. So it's introduced by infection of the bacterial lawn on an agar plate. So the culture, the growth of viruses is made more difficult than the culture of bacteria or fungi by the fact that viruses will only grow. Now what you do, you're placing the gene in the vector. So you have the plasmid DNA. The DNA molecules are small and can be easily separated based on their size. So bacterial cells are broken and the chromosomal DNA is removed. It leaves the plasmid DNA to the pellet. Now the plasmids are purified before cutting it with a restriction enzymes. It's very simple. Now you add the vector, you add the DNA, and it generates sticky ends. And due to the hydrogen bonds and complement DNA strands, it will come and sit. But for joining it, you need the phosphate backbone on the phosphate at the 5' to be joined with the 3' OH using the DNA ligase with the help of ATP. So that's the very simple way of telling. Now the other way around is like how do you okay, so now you have to get into the transformation. So this is in the simple words of how you do it. Now for the transformation, you have the bacteria such as E. coli that can take up and express the foreign DNA in the form of a plasmid. So the clone gene is inserted in the plasmid in the bacteria, and it's led to grow. So for that you need to first choose a bacterial host, that's the model organism. Like in E. coli is a good model organism because it's well studied. No nuclear membranes, has enzymes necessary for replication, grows rapidly, It's inexpensive, normally not pathogenic, and it's easy to work with and transform. Now, for the steps of bacterial transformation, you have to choose the plasmid to transform. For your case, maybe it is the plasmid that is already present over there. Then, so the plasmid is that has a single recognition site for cutting it at places. It has an origin of replication from where the replication procedure starts, and then the marker genes. Okay, so like I was telling, there is an antibiotic resistance and the one that expresses color. So the bacteria is plated on a media with an antibiotic that it's resistant to, so only the bacteria that have taken up that plasmid will only grow. Now, bacteria that have taken up the recombinant plasmid are of a different color than the bacteria that have not taken up, so there is a color generation. So these are characteristics needs to be present in a particular plasmid. So the steps for bacterial transformation is like you treat it with calcium chloride, or you undergo electroporation and then directly inject the plasmid. Then you grow it in certain nutrient conditions like LV agar or something, and plate it on that agar plates and let it grow overnight. The next day you come and observe the plates. Now the other thing that you need to do is the PCR, that is the polymerase chain reaction for the amplification of the genes. Okay, now for the amplification of the genes, what you need to do is like, so this particular reaction was first described in 1971 by Clip and Corana, and then later remodified in 1985. So it's a very fast, powerful, adaptable, and we call it simple, but it's not that easy because there are certain factors that needs to be checked before doing it. But it has many applications. So why do we need to amplify sequences? Mainly for cloning, sequencing. We need a lot of DNA material to verify the identity of the engineered DNA constructs, to monitor the gene expression, to diagnose a genetic disease, to reveal the presence of microorganisms, then to identify an individual, etc., so on and so forth. Okay, so what do you need for PCR? So you need a template DNA, that's the target sequence that you need to amplify. You need the primers, that is short oligonucleotides that define the ends of the target sequence. You need a thermostable DNA polymerase, buffers, DNTPs for adding it to the different complementary sites, and a thermal cycler that will change the temperature from time to time. Now how it works is basically first you denature the template strand by adding high amount of heat, like from 94 to 98 degrees, and then so the strand opens up, and then you allow the primer. So you have to make primers. So primers are short segments of DNA, like about 20 to 25 base pairs, and that comes and sits in this position. So you have to make two primers. One is the 5' to 3' and the 3' to 5' primer. So it will come and sit, and then the DNTPs keep on adding in these portions, both on this side and this side. So from one particular strand you get two strands, and then you have to elongate it. So there is elongation at the optimal temperature of the DNA polymerase, and then what you do is that, so after one particular round you get, if you start with one template DNA, you get two template DNAs. So now in the second cycle, the similar things occurs where you have denaturation. So you can get, like, after round two you have four. So from there you will, after at the end of, like, you get four strands, from where four double-stranded DNA will be prepared. Okay, so that is how the process continues, and by the end of like 29 to 30 cycles, you will get 2 to the power of 30 DNA strands. That's a huge number. Like, you can see the number over here. It's basically very huge. Okay, so usually we do it for 30-35 cycles because we not need a large amounts of DNA. As long as you have the template, as well as there is your DNTP, or the activity of the DNA polymerase does not gets affected or utilize, its becomes not active, it can go on with the amplification. So like, it's an optimized of 30 cycles usually. So that is all about PCR. So PCR is very, very important because you have to amplify the insert the genes to carry out the different kinds of functions even when you're inserting it in a particular vector, right? Okay, any questions? If you have any questions, you can go ahead. Okay, so if you have no question, I think we can go with the question answers. Not very complicated though. We should be able to answer them. So, like the first question, maybe. So, the first question says plasmids are successfully used as vector for gene cloning as their extrachromosomal small circular DNA. Double-stranded DNA. They have an origin of replication. They possess selectable marker, or all of the above. Yes, so it's option D. Option D is the correct one. All of the above. The next one says to cut a DNA molecule, which of the following enzyme is used? We have studied about it. Is it the ligase enzymes, restriction enzymes, phosphatase enzymes, or the polymerase enzymes? So these are the restriction enzymes. That is question number 2, option B. Okay, restriction enzymes are used for cutting the DNA molecule. The third one says the DNA molecule in which gene of interest is inserted for transferring to another organism is called. So converter, vector, exporter, transporter. It's simple like a vector. So vector is the one where you insert the particular gene of interest or the DNA sequence that you want to actually study, or you have engineered. Now to join gene of interest, so question number 3, it is B. For question number 4, it says to join a DNA gene of interest with the vector, which of the following enzyme is used? Ligase, lyase, ribonuclease, and the polymerase. We know it's a ligase enzyme. So ligase enzyme joins the 5' phosphate with the 3' OH with the help of ATP. So ATP is very, very important as it provides the energy. So option number 4, it's A. So question 4A. Now the mechanism of intake of DNA from surrounding environment is called, so what you do is it conjugation, transduction, transformation, and gene cloning. So we know about this technique. So you are inserting a foreign DNA from the surrounding, so that is a transformation of the bacteria. Transduction and conjugation is very different because that requires another set of processes and all, right? Now, the role of a selectable marker in a cloning vector. So, you need a selectable marker. Why is a selectable marker required? For maintaining the vector in cells, selective survival of the cells within the vector, increasing cell growth or expression of the gene in the cell. So, a selectable marker is definitely used for selecting the survival of the cells that have that vectors or marker is generally required to understand whether your insert or the plasmid has inserted into the bacteria or not, as well as it is very well important to understand the um, whether your cloning has also occurred or not, like if there is a change in the color, and also you require a selectable marker. So one commonly known selectable marker is the antibiotic resistance that is present within the plasmid. So here the two answers are actually correct. One is the maintaining the vector in the cells, and the other one is selective survival of the cells that have the vector. So question 6, it is both A and B. Multiple cloning site or MCS in the vector has promoter site, ligation sites, restriction sites, and resistance sites. So cloning sites are the positions where there are a set of multiple palindromic sequences, each represented by different restriction enzymes. So MCS contains like maybe 20 to 25 restriction sites, unique restriction sites for chopping it and inserting your different inserts. What will happen if the original, so this question number 7, it is option C. What will happen if the origin of replication is missing in a vector? So origin of replication is important for the vector to replicate. So if this particular sequence is missing, there won't be any problem for taking up by the cell or for containing the gene of interest, survival and all that won't matter. What it will do is basically the vector will not replicate. So option D, that is question 8, option D. A large number of copies of gene can be obtained following. So you need to obtain a large copy of gene. What you need to do: transformation, polymerase chain reaction, plasmid isolation, or whole genome extraction. So like I was telling, for the amplification, large copies of genes means you need to go for a DNA amplification. So if you need a DNA amplification, you need to take the help of polymerase chain reaction, because that is the only way you can amplify them by in like lakhs and crores of genes can be created. So the option number 9, it is question 9, it is option B. For question 10, which of the following statement is true? PCR machines are known as thermocyclers. That is correct. Exponential amplification of the gene can be done from a very small amount of DNA. That is also correct. Thermostable DNA polymerase is absolutely necessary. Yes, because why thermostable it is necessary? Because you are heating that particular mixture of template, that particular PCR mixture at 98, like around 100 degrees. Okay, so if the DNA polymerase is not thermostable, then it will just lose its activity at that high temperature. So for question number 10, A, B, C, D, all of them are correct. Or if it's a so D is the more correct. So question number 10, it is D. The 11th question says the first step in PCR. We all know it starts with denaturation, then go for annealing, extension, and finally there is the renaturation of the entire thing, and then it goes into this round 2. So for question number 11, it is B, that is denaturation. To which side of the primers the restriction sites are included in cloning? It's a very, very interesting question. So where do you add the restriction sites for a particular primer? So according to our knowledge, how much we know that there is extension at the 3' end. So always we need to keep the restriction site at the 5' end, because we do not want to hamper the insertion of that gene or do any kinds of activities in the middle of the gene by inserting the restriction site. So it is always at the 5' end of a primer. Okay, so the restriction sites are always put it, be it a forward primer or a reverse primer, does not matter if it's a restriction site because it's palindromic, so it has to be in the 5' end. So the stop codon is present in the 5' end of a forward primer, 3' end of a forward primer, 5' end of a reverse primer, or the 3' end of a reverse primer. It's a very, very interesting question. So if you look into a particular template, it all, or how does a protein produce? It produces from the 5' to the 3' end. We need to just track back a little bit. So how do we track back? So for tracking back, we need to understand how will the mRNA sequence look. So it observes the 3' to the 5' end, and it creates a 5' to the 3' end mRNA. So if we look into the 3' to the 5' end, So 3 prime to the 5 prime end is definitely for a reverse primer because we think it in that manner. It has the same sequence as a reverse primer. The 5 prime to the 3 prime end will be the forward primer. So we need to imagine by the reverse primer. So the 3 prime to the 5 prime end. So if when the mRNA starts, it will start at the 3 prime end of a template strand. Okay, so from 3 prime end there has to be like the complementary of ATG, that is cat or something, so it will start like ATG. Okay, so then it will start making the 5 prime end, and we want the mRNA to have the stop codon at the 3 prime end. That means for a mRNA to have the stop codon at the 3 prime end, we need to have the stop codon at the 5 prime end of a reverse primer, right? So the 5 prime end of a reverse primer will have the stop codon, and like the previous questions it asked, where should be the restriction enzyme? So like you should have the gene, you should have the stop codon, and then after that you have the restriction enzyme, restriction site. Or if you look it from the 5 prime to the 3 prime end, it's the restriction site, stop codon, and the template sequence in the complementary format for a reverse primer. So it is option C. For question 14, it says the PCR, the DNA synthesis. So DNA synthesis takes place at. So DNA synthesis means it's the extension stage. So extension stage means where the DNA polymerase, the thermostable DNA polymerase is highly very active. Okay, that is at 72 degrees because that's the temperature where the DNA polymerase are usually very active at. The last question says expression vectors differ from cloning vectors by having. So is it a selectable marker? No, that is present in both the expression and the cloning vector. It's the origin of replication. No, it has everything. And the gene expression control elements, yes, that is correct, because a cloning vector cannot be used for the expression of a protein, but an expression vector should have those qualities. Why is it so? Because a cloning vector is for maintaining of a plasmid where it has Reke Inde mutation. So what are these mutations? It does not allow. So Reke mutation is recombinase A mutation that does not allow the recombination of the DNA because you do not want the DNA to change over time. So that is how you store or preserve your plasmids. And the Inde mutation is the endonuclease A mutation that does not allow for the cleaving of the DNA over the course of time. So therefore option B is more correct. So question number 15, it's B. Okay, so yeah, that's all I have. You guys have any questions? Like, no questions. Okay then. Yep, thank you very much. We will meet again in the next week on Friday.